right, welcome folks to another week of the Bioinformatics workshop series. This is week 7. We are almost done, and we are going to be taking a little bit of a distancing from what we have done so far, so a little bit less hands-on this time. We're going to be talking about what some of the current applications of AI and ML in the world of informatics, bioinformatics are. First, before going into a couple of examples, Carlos is going to talk to us a little bit about what AI and ML are. And then we're going to see a couple of examples of how these are being applied in the world of bioinformatics. Let me send you the link for today's materials. Here you go. Feel free to follow along. As I mentioned before, today is going to be a little bit more of a conversation style where Carlos and I are going to be having a conversation, and you are very welcome to give your input regarding things that you may have to say on the topic. Okay, I'm going to stop sharing and Carlos can take over.
And my audio. Okay. So now everyone can hear me? Yes. Okay, so we start going to review some of the basics of what is machine learning, and also we would like to make a difference between machine learning and deep learning. So machine learning can be a general term, and then if we want to focus only on specific type of algorithms, then we can talk about classical machine learning, and deep learning will be related with something else like neural networks. So there's always a mix of different fields. And we have here what is bioinformatics, we have here what is information science, and then we have here AI, artificial intelligence, and then a subset of the artificial intelligence is machine learning, and another subset of machine learning is going to be deep learning, and this is how it's a combination of bioinformatics, information science, and data science. Okay, so and we know, for example, all about AI. AI, well, is used, for example, for doing speech recognition, decision making, self-driving cars, and also it's applied in healthcare for example, trying to automate some specific image detection algorithms that classifies different images, for example. And we do in machine learning, for example, we can try things like studying protein structures or do disease diagnosis using specific algorithms. And then when we're doing deep learning, then we will involve special algorithms that are called neural networks to process different data. Okay, so we continue. We can do an exploration of what is classical machine learning. So classical machine learning is related with two types of tasks, those who are supervised and those who are unsupervised. When we are doing supervision of the data, so all the data can be numerical, and also we can use labels. And if we are doing labels, then we can do two types of tasks. One will be classification. For example, if you have in your home set of socks, then you can separate your socks by colors. So that's classification. You can say white socks and colored socks. And if you have numbers, then you can do prediction. For example, what is the length of the ties in your house? So you can order your ties by size and then predict what size you don't have of ties. On the other hand, if you have unsupervised data, so the data is by itself. For example, you have images of cats and dogs, and then you can do clustering. So, for example, here you can split up similar clothing into stacks. So you have different ones. You can have pants, or the other set will be shirts, etc. And then you can do dimension reduction, which, for example, what's the best outfits from the given clothes. So you go to your closet and then try to select something. And then, for example, if you want to do a more in detail association of your clothes, then you can say, well, this pant goes with this shirt and these shoes, and then that is an association. Find clothes that often wear together. So those are the types of algorithms you can use in classical machine learning, and basically you can do classification, regression, clustering, dimension reduction, and association, and some other. Okay, and also you will find specific topics like what is linear regression or logistic regression. So all the regressions are just trying to predict a number. And then you have decision trees like and random forest and support vector machines. These are for using classifying things. And at the end, you will have neural networks, which are more complex. Okay, you will find here a link to our notes of what is an overview of machine learning, and that you can explore. Maybe I'm not going to go into it. Okay, the other topic besides classical machine learning, then it's deep learning. So what is deep learning? So in deep learning, we will find that frequently people are talking about neural networks, and basically the idea of a neural network comes from biology, where you have dendrites and axon, and you have all the different signals that travel between all the neurons that are connected. So similarly, if you're doing we can have a system that we have an input layer of data, and then this data can be like signals. They will go, they will travel to the right, and then at the end you will find some action, and this will be the result of all this input layer. Similarly, as you have all your neural networks inside your body, where you have some sensation, and at the end you have some feeling, which is the information processed by your brain, etc. So those are the similarities. And in neural networks, you always find an input layer and an output layer, and in between, you will find the hidden layers, okay? And basically, the idea of a neural network, I'm going to open this link. And this is the basic idea of a neural network. You have several inputs, and then you have some function, and then you will have an output, and maybe you have some correction number. So I'm going to present here the mathematical expression for a neural network. You have an input, and then you have something that is modifying the input, and you will have an output. And mathematically, it will be just like a linear equation where you have your input, which is x. Then you transform x with some function w, and you will get the result y, and which is the same figure as this, okay? And this is a linear equation, and we are used to linear equations. For example, when we have a plot, for example, we have some data. So we have 3 blue dots, and what we want to do is try to predict more values. So what we do is just a linear regression. or we are just feeding a line that goes near the 3 points, and we always can find the best line or the best fit that fits these 3 experimental points. And the best fit will be that one that the error is minimum. So there's an optimization problem. Okay. And when we are feeding a line, I can just go directly to here. When we're feeding a line, there's several ways we can fit a line. So I can play with the parameters. For example, I want to have a best fit of a line that goes through these 3 points. And if the line has different slopes, I can make my error will be bigger or smaller. And then this is the error, for example. Then I can play with the bias, which means the intersection point with the y-axis. So the idea is try to optimize a nice slope and the less error, or sum of the squares of the error. So this is, for example, my error is 688, which is better than the other value of 750. So this is one equation. And geometrically, you have the initial point, you have the application, the transformation, and then you have the result. Okay, and then similarly, you can have several things going on at the same time. So you can have multiple inputs and different functions and different bias to get the output. So that's the idea. You can generalize instead of just trying to fit a line, you can fit a plane in a 3-dimensional space, etc. So that's the idea of a neural network. We're not going to go into too much detail. And then neural networks can be several types, several ways of having an input, some transformation and an output, and between the information between the different input layer can be connected through the different hidden layers, and then you have an output. So depending on the type of applications you have, probably you will find that there are some neural networks that are highly used for specific problems, like the generative adversarial networks or the recurrent neural networks or the convolutional neural networks. So there are several types of networks that you can use in order to solve a problem. So the best thing to do is when you're reading a paper and people are using neural networks, see what type of networks they're using and work from there. So in general, when we talk about machine learning or artificial intelligence, we will try to classify things between 2 things: the classical machine learning, which is supervised learning and unsupervised learning. And then these are the types of tasks. We can do a regression, like fitting a number, or classification, or we can do clustering and factor analysis. And you will find all these different type of algorithms available for doing regression. For example, you can do a spline, you can do a nearest neighbors, or XBoost, or classification to the logistic regression, the support vector machine, random forest, etc., clustering the k-means, etc. So you will find different, in factor analysis, you will find, for example, the principal component analysis and others. And in deep learning, you can do, you can analyze unstructured data like language or text. And also you can do time series analysis from different sensors. For example, all the sensor data that you are picking up from a specific device. And there are some other types of learning, but we're not going to address them here. So what is the difference between classical machine learning and deep learning? In classical machine learning, you have your data, and so this is the left-hand side graph belongs to the classical machine learning, and the right-hand side belongs to the deep learning. So in classical machine learning, you have your problem, you have your data generation, you will have raw data, and then you start pre-processing, for example, eliminating empty values, eliminating outliers, or even scaling the variables into, or normalizing your data variables, etc. And then once you have your pre-processed data, then you build your machine learning model. You have a hypothesis, and also you start trying to validate the performance of your machine learning by playing with different hyperparameters until you get some accepted predictive model that has very small error. On the deep learning side, you just have your data. You give your raw data into the model. The model will learn all the different relationships within the data and can just give you an output. And you can have different metrics for doing deep learning, like the mean square error and other types of metrics to see if your model has the correct number of layers and the correct number of parameters in order to have a good predictive model. So that's basically the difference between classical machine learning, where you have to intervene in your data, and deep learning, where you don't have to intervene. So you just give the raw data, and the model by itself will try to determine what is the output. And you will find different types of models, like the perceptron, or the multi-layer perceptron, or the convolutional neural networks, transformers, and now the most popular things are the large language models like ChatGPT. And this table gives you a difference between working with machine learning, classical machine learning, and deep learning. And you will have different types of algorithms. You also, if you're doing machine learning, you can work with small amounts of data, but if you're doing deep learning, you need large amounts of data, like sensor data. Classical machine learning are typically fast, and in deep learning, you need more computational infrastructure. In classical machine learning, you can play with your data and see what variable is more relevant than others. In deep learning, it will learn by itself. And if you're working with classical machine learning, you need to prepare your data or pre-process your data. And on the deep learning, you just keep the raw data. On the fine tuning, you can play with, in classical machine learning, you play with the different parameters in the hyperparameter space, trying to optimize the performance of your model. And on the deep learning side, well, everything can be learned by the algorithm, right? The complexity in classical machine learning, you have simple mathematical models, while in deep learning will depend on the architecture of your neural networks. On the classical machine learning, typically the process is transparent. You know how your data went in and what you modify or optimize in order to get a good performance. If you have deep learning neural networks, it's just a black box. And in classical machine learning, you can explain the outcomes, but in deep learning, it's hard to, for example, once you have an output from ChatGPT, it's very difficult to trace down why the model is giving you that answer. Okay, and then we will have all these different learning resources where you can learn more about machine learning, more about classical machine learning, what type of algorithms there are, what type of libraries in Python exist for working with classical machine learning, and all the different types of neural networks, and that's about it. Okay, Michele, if you have any questions or...
No, you can stop sharing. I can share on my side.
Okay.
Okay. So thank you, Carlos, for the introduction to machine learning. extremely useful to understand that nowadays we have access to this technology, and because we do have access to this technology, we need to also understand what it is and how it works. So what I'm going to do now is that I'm going to give a few real-world examples of how this technology is being applied to bioinformatics. Now, it is not the easiest to find some papers that you can understand if you do not understand machine learning, but honestly, between you and I, there is so many resources out there that can help you understand some of these papers. I'm thinking of the chat GPTs. Nowadays, another one was released called Notebook LM, which allows you to upload a specific PDF, and it will create a small little summary about what this of what the source that you're uploading is. So I've selected a couple of papers and also a couple of tools for us to look into. Now I can share my screen. So MLAI has ascended this data discovery, the data discovery of the data that's all around us. We can literally go from the microbiomes that are on these tables right now to how many molecules there are in the air. And before having access to ML and AI, all of this data collection and data extrapolation will take years, if anything. So MLAI has definitely accelerated this this process of taking data, making sense of the data that of everything that is around us. Another thing that we do need to take into consideration is that ML and AI require a lot of resources. First, it takes resources to run these platforms to create the models, and then the models themselves. Sorry, first it takes so much space to uh, run the clusters and on your own machine to create to use the model data, and then it takes forever for us to actually create the models themselves. So by no means today is supposed to be something like you can go home and do ML on your data. Not necessarily. So this is going to be more of an expose of what ML and AI is being used in bioinformatics. One thing that I do have to say, however, is that I've taken a couple of papers from papers with code, and if you have never used papers with code, is a great resource. And let me show you why. The name is almost self-explanatory. This is a website which loads up papers or academic papers which also come with code. I can type in machine learning statistics, and I want to see what comes up. Nothing comes up. Fantastic. Let me try to stat. Oh my God. There we go. And all of a sudden we do get a few hits. The first one, I'm not going to go too deep into it. Learning phrase representations using recursive neural networks encoder decoder for statistical machine translation. Let's look at it a second. Papers with code will give you the blurb of essentially what the paper is about. You can go ahead and look at the PDF of the paper or read more on the abstract. Usually this will lead to an archive. I think it's the RXIV, yes. So if you want to read more on the paper, you can go right ahead. If instead you're just interested in running the code, Papers with Code also gives you the direct link to the paper's repository. And through here, you can skip the paper, and just go ahead, download the code and trying to see whether you can run it yourself. So gonna close this up because I did want to mention that this is a resource out there. The first paper that I wanted to kind of look over is machine learning algorithms to infer trait matching and predict species interaction in ecological networks. I'm putting this image right here, but I'm going to come back to it in just a second. And essentially within this paper, what the researchers did was to compare different machine learning methods, the one that Carlos did talk about earlier. These are the more of the classical machine learning methods. So random forest, boosted regression, trees, deep neural networks. Well, deep neural networks, I think it's more of a non-classical machine learning, same as convolutional neural networks. But support vector machines, I think it's more classical as the k nearest neighbor as well. And they essentially, what they were trying to do was to observe interactions between, well, nature itself, seeing whether there was any, where was it? Whether there was any statistical relevance or I'm scrolling up and down over here, but I should probably just go to the paper itself. Once again, if you do want the code, you can just go right into papers with code and open up the code, the GitHub. So they were looking to see whether there were some interactions between pollinators and their various flowers. And obviously, this could have taken forever to really make inference. So they decided to just give all of this data to a number of machine learning algorithms to see whether it would make sense for the machine learning algorithms. And what we figured out is that, well, yes, we are able to extract more data by feeding it whatever algorithm you may want to use. This study itself was more of a comparison between each of the methods themselves rather than a single, oh, go ahead and use this method is better than everybody else, than everything else. And the image that I was saying that I was going to come back to earlier pretty much takes every single one of these methods and compares it against each other in some sort of a clear way. It shows you in the dotted lines what data was used for training and then what data was used for validation itself. And then it compares it between accuracy, precision, specificity, sensitivity, and true skill statistic rescaled. I'm not sure exactly what that is, Carlos, if you are aware. No. Thank you very much. Oh, well, don't worry about it. And area under receiver operating characteristic curve. Now, I'm not going to go in. I don't have the details and knowledge enough to really understand what all of these are, but at the end of the day, we can kind of see that the shape of all of these machine learning classical methods, it's pretty similar, which means that if you are using any of these methods for your own research, you're in safe hands. But what did this research, this paper really figure out? Well, that there is a relationship between excuse me, I'm scrolling down to my notes, that there is a relationship outside of the species themselves, as in they are moving beyond the phylogenetic proxies, where previous studies often relied on phylogenetic information as a proxy for understanding whether a specific animal will go to a specific flower, for example. But using machine learning, you can kind of understand that there are other interactions outside of the phylogenetic proxy itself. And what machine learning also helped with this is to also figure out whether how the ecosystem responds to the specific change between all of these interactions and understanding the mechanisms that go throughout a large ecological area. I liked this paper because it was a good introduction to the question, what machine learning algorithm should I use for my research? And I think the answer was it doesn't really matter. They all score somewhat similarly. Um, apart from SVM, which I forgot to make a note of. Support vector machine. There you go. Support vector machine. If you're looking for more accuracy, you may want to go for the KNN, which is K nearest neighbor. But it is really up to you which method applies for your own research. Next is the large-scale machine learning-based phenotyping significantly improves genomic discovery for optic nerve head morphology. It does matter if explainability, excuse me a second. It does matter if, thank you. It does matter if explainability is important to you. Randall, you do have a point. Thank you for pointing that out.
There is a term which is called ML explainability.
Mm-hmm.
So the idea is that once you have your classical machine learning models, you will have the feature engineering seeing what is the result composed from, for example, let's say you're studying some problem that involve gender, profession, age, nationality, and then, for example, some variables will have more importance or relevance than other.
No, you both bring up a good point. Thank you for explaining that, Carlos. The comment from Randall in the chat right now also makes sense. Definitely it's true if you require explainability. So yes, thank you, Randall, for pointing that out. I guess this specific study did not really take that into consideration, since it was just looking at, I will say, matching all of these machine learning algorithms against each other to see which one works better. Can you let me know, Carlos, if any of these are better at explaining rather than others?
No, they're just different metrics.
They're just different
For
metrics.
example, the area under the curve will tell you something, the specificity or the precision or recall or confusion matrix will just give you different metrics and trying to compare one model versus the other.
Okay.
And also you should always have several models, not just one.
That's fair.
And then take a voting.
Take a what?
Take a vote between the different models if you're trying to predict something.
Okay, thank you for that. Okay, yeah, the next paper that I did look at was, well, this really long name, and essentially what they tried to do was to use machine learning in order to predict the glaucoma-related traits from color fundus photographs. And if you do not know what color fundus photographs is, this specific image here, where you look at the eye in, you take a picture of the inside of the eye, essentially. They were able to train a machine learning algorithms using 81,830 photographs, which is a lot of photographs, if anything. And what they were trying to do was to automatically predict VCDR, which is vertical cut to disk ratio. Now, I'm not a medical doctor, so I can only say that is this specific area inside your eye. And apparently, if you do have glaucoma, this changes or morphologically changes over time. Making this a little bit further smaller. So out of 81,830 images, this was split into training, tuning, and testing. And they ended up creating a model. Now, I do not remember if I have written here what algorithm they did use. Gonna go ahead and dig into it in just a second, but then they were able to use their model and look at further 175,000 images and well, essentially try to infer a genome-wide association study using their VCDR predictions. They were able to find, they were first of all able to very quickly identify whether a certain person was affected by glaucoma. And also, they did run into some issues where they weren't able to find, they weren't able to categorize a specific photo. But they were able to infer 92 novel loci based on this massive study that they did with 175,000 images. Hold on a second. I do not remember what algorithm they had decided to use. Let me see if they mention it very early in the abstract. I do not see it. I'm gonna go down to move to methods, unless you're ready to. Gotta move down. Okay.
ML-based GWAS. I don't know what.
Maybe that's why I didn't look to. I did not make it a note of it. Excuse me a second. I'm gonna go look at the code itself to see whether it does mention it over there. And that's one thing I like about papers with code. You can jump between one and the other. Uses Python. Oh well, I'm pondering on this too much. Just gonna go back to the nodes themselves. A final thought about this is for sure they used a lot of images to train the model, but using that model to then run a genome-wide association study with all of that data, they were really able to figure out 91 loci, which is pretty hard in in the world of bioinformatics through a single study. So congratulations to these folks. And now I kind of wanted to switch gears a second and moving away from papers and talk about two software that are extremely powerful and do use machine learning methods in order to function. And the first one is AlphaFold. I myself am not a person that works with proteins. So if you are working with proteins, do point out wrong things that I may say. But first, who won the Nobel Prize this year for chemistry? And the right answer, or at least the partial right answer, is Demis Hassabis and John Jumper. Demis Hassabis is also known to be one of the founders for Google DeepMind, and that is what AlphaFold actually runs on, or it is trained on. And what AlphaFold does is that it is able to predict the 3D structure of proteins from just their amino acid sequence. It's incredibly powerful. I've had the pleasure to work with AlphaFold 2, or at least see people work with AlphaFold 2, and it's just mesmerizing. And now you can use AlphaFold 3 straight from your browser. But before we do that, I just wanted to show you how AlphaFold works. So it does take in a protein amino acid sequence, and then it runs something called a multiple sequence alignment, or an MSA. And it compares this specific alignment that you have inputted onto a massive bank, essentially. It then runs through its own neural network architecture in order to create the protein and to create the prediction and the structure of the protein itself. Now, you can try this out for yourself. If you click on AlphaFold, I'm gonna copy this and send it over to the chat. Oh, thank you for the discussion, Carlos. And this is the AlphaFold free server. You may need to register for it, or you can use your Google credentials to get into it. And I can't see because of Zoom. And you can click straight on the server itself, where I already have loaded a sequence right here. It's one of the sequences that they have provided, the RNA ion sequence. And if this is not your first time looking at protein sequences, this is what a protein sequence looks like. It's a bunch of amino acids put together. And you can click on continue and preview my job. Now I'm not changing anything around, but I can go ahead and show you what it looks like. Job successfully launched. Make it a little bit smaller. And I can click here at the bottom. Once it is done, here is the predicted protein. This was not pre-rendered or anything. This is created in real time, or let's not call it real time. This was created using AlphaFold as I have submitted the job. And it gives me a number of extra information that I'm currently not able to access, I believe. And this other extra information that I wanted to talk about, you can find in the AlphaFold protein structure database. Copy this real quick onto the chat. And this is a massive database of all protein structures that are created by AlphaFold and submitted by people around the globe. Guess I was not able to find that same protein. Let's try that again. Aye, aye. This is probably not the same protein, or not that I know of, but it does do something similar, which is allow you to view the protein itself and give you some extra information about the model confidence, for example, similar structures, which it does not load at the moment. Here is another example that I have pulled up. You can find similar structure in other similar proteins and different species, perhaps. In this specific case, we're all in one species. And in also what other, I'm not sure what these are. Well, other species, but other types of the same protein. I don't want to say things that are wrong. I just do want to go back to AlphaFold where I can say it's incredibly strong and it uses Nobel Prize technology right behind it. So you can go ahead and play around with it. It's incredibly powerful to just witness. And the last thing that I did want to mention is deep variant. And deep variant uses, first of all, it's from Google, and it uses convolutional CNN, so convolutional neural networks, in order to infer variant data. And it does this in a way that I find extremely impressive. So essentially, I think I have it written down somewhere, but what it does is that it takes the BAM file, a BAM file that we have talked about for so long, and it transforms it into a matrix, a two-dimensional matrix. And then you can take every other BAM file that you have and transform those into a matrix to then create essentially what is a data cube which is then fed inside this really powerful convolutional neural network, and it infers whether there are variants within this specific data cube of BAM files. I'm calling it a data cube, but it's actually called a pila, and I find that extremely smart. I don't know if something similar is being done in either of you guys' field, but this feels very much like it's the first time it's being done in the field that I've worked on.
Like my field directly uses images,
Right.
which are two-dimensional arrays, which is specific, what CNNs are good at. But I was so it's in this case, deep variant is taking genetic sequencing and turning them into an image.
Image, yeah.
Image, and then do a CNN. And so that makes sense, like the way they put them, like at the order of them. Apparently that must mean something with the genetic variants.
I've personally never used it, but I suppose that if I were to try it out, I will show you because this will be interesting to see, witness.
Yeah. Taking data and turning it into an image, right? And then apply CNN to.
Exactly. Eric says, when in doubt, make a structure of an image, literally. It has to be said that deep variant was trained on extremely large data sets. Google does have the money to do things like that. So, well, thank you, Google. I suppose I want to try it at some point going forward. I tried to also understand a little bit about tensors and why is it so effective? Well, that's because it creates this massive pileup of images and it inserts it into what we call a tensor. A tensor is essentially a generalization of vectors and matrices that can be modified into higher dimensions without losing the, oh my God, the resolution, right? Or am I wrong? Like you can expand the dimension of a tensor without losing the resolution of a specific vector.
Yeah, these are vector images.
Right.
It can be scaled or compressed or stretched.
And I find that incredibly smart.
It's just a mathematical equation.
I don't come from that side of I don't come from that side of science, so it's insane. And it then uses the 3 dimension of this pileup cube to maintain the information of the genetic information of the genome itself. The first dimension is the height, number of reads at a particular position. The second dimension is the width, corresponding base position alongside the reference genome. And the third dimension contains the channels, which are quality scores, mapping quality, and strand information. You got it. Okay, cool. So Google Deep Variant, I find it extremely interesting. Oh, Randall's got something to say. When you transform a genetic data sequence into an image, do your color code for characteristic SCTG so that you feed a color image into your CNN? I would love to know that, Randall. I'm not going to lie. This was a pretty recent discovery from me. For me, you can. I'm gonna send you the link to their GitHub, which has a set amount of case studies for you to look at, and I think it will be there is definitely more to it that I myself do not understand. Also, excuse me a second, I would like to stop sharing because there is a link I want to send you, but it's in my messages. And oh no, it's right here. What am I talking about? This is a recent paper called Deep Learning in Bioinformatics, and it does cover deep variant as well as other deep XXX tools such as Deep Trio. If you look into the paper, scrolling down. Ooh, scrolling down, as you can see, there is a couple of images that we have used for today. Deep Gene, Deep CPG, Deep Bind. There is so many that I have also not have heard of as well. So I'm not sure how it actually works. So Randall, excuse me, I can't answer your question, but if you're hopefully one of these papers is able to help you with your own research. And Eric says, pretty nuts. Yes, it is. And folks, we are at the tail end of today. Sadly, I do not have much ability to answer your questions. My field is not necessarily machine learning, but there is so much out there that's worth learning, and there's so much out there that's worth discovering. So by all means, yes, I wouldn't say that it works on color. It does work on numbers. So the image classification works on color images, doesn't it? It works on numbers. When an image is being looked at, the color is not looked at, but the numbers behind the color is. That's at least my understanding of it. And everything ends up in being numbers. So perhaps when we call it an image, it doesn't necessarily become colored, but each
RGB
Yeah, three
combination numbers.
right,
arrays, red, green, blue, and number.
right. But it doesn't have to be colors, right? It can be something
It's
else.
just numbers.
Okay.
But
Can be
red,
converted
green,
to
and blue have their
grayscale.
own array of numbers.
Yeah, my colleagues over here are saying red, blue, and green, but what I'm trying to argue is we can look at beyond those and just look at the value of the numbers themselves. But those are red, blue, and
It's
green.
just
Okay. Yeah. Okay.
three of them for a color.
Okay. So Randall, maybe. Or grayscale. There we go. Yes. Folks, we are at the tail end of today, and with 4 minutes to go, once again, I would like for you to please fill in the feedback form. If you have any feedback to give, it only takes a minute to give me feedback. And if you have any questions, please feel free to reach out.
The feedback link.
I did not put the feedback link. Oh, I put the wrong link in. Sorry, shortened. Here's the form. There we go. Next week, we're going to have a guest speaker for this series, Artin Majdi, which is going to actually try to connect some of the dots that we have discussed today about machine learning. Earlier in the series, we talked about workflows, and today we talked about machine learning. Machine learning and workflows don't always go hand in hand, but there is a specific tool that does it quite well, and that is called ML flow. This is a little bit outside the topic of bioinformatics, but it's essentially a tool worth knowing if you're going to be doing any sort of machine learning work. And that may also include machine learning in bioinformatics. So by all means, I hope that you all do come next week. And thank you all for your questions. And Randall, feel free to straighten me out if I do say something wrong. With all that said, thank you all.